So this is an exciting transition for the course. At this part of the course, we actually move on to true metabolism. We'll be setting the stage right now for the concepts that we will encounter as we move on to the metabolism of glucose, but this really marks the beginning of that journey. It's about a 8 to 10 lecture journey that we take to go through all of the metabolism of glucose, especially if we include photosynthesis. And it starts in this lecture series. So this is the lecture series for lecture 9, the first lecture of the second unit of this course. There will be 3 chunks to this lecture, 9A, 9B, and 9C. And this is chapter 15 in the textbook. Essentially, we'll be talking about redox reactions and how cells use redox reactions, or to say it more simply, how cells move electrons around as a mechanism for moving energy. So in this first chunk, we'll talk, first a little bit of a digression. We'll talk about the standard states for chemical reactions, just as a definition so that we know what that means. And then we'll set the stage for metabolism itself and just define some terminology and make sure we're all on the same page by what we mean by metabolism. We'll begin the next chunk by talking about redox reactions themselves, introducing the concept and explaining what goes on in a redox reaction. And the last chunk will focus on coenzymes that help enzymes engage in redox reactions and the coupling of reactions to drive positive delta G reactions forward. Remember, positive delta G reactions should never occur, but we can actually play a biochemical trick where we drive positive delta G reactions forward by coupling them to reactions that have a negative delta G. So we'll talk about that in the last chunk as well. Oh, and coenzyme A will be the last thing we talk about in that chunk as an example of activating an otherwise non-reactive molecule. So I think our textbook is great. I've been using this textbook for a long, long time. I think it's clear, it has wonderful figures, it's easy to read, or as easy to read as a biochemistry textbook can be. But one thing that I like about this textbook the most is its organization. I think the authors have done a really good job of ordering the content in a logical way. And that's especially true right here for redox reactions. So redox reactions, conceptually, are critically and centrally important to metabolism. If you don't understand redox reactions, you're not going to understand the metabolism of glucose. Redox reactions are essentially the energy harvesting reactions of life. So these are the reactions we use to make ATP. Obviously, if there's no ATP, there's no life, so it doesn't really get any more important than these reactions. But they're complicated. There's a lot going on, it's a little bit abstract, much to keep track of, and students sometimes struggle with redox reactions, especially when those reactions are brought up in the context of metabolism itself. So you've got the chemistry going on, you've got the energetics going on, and then also on top of all of it, you have the redox reactions going on, and it's a lot to process, understandably so. So our textbook does something very ingenious, which I agree with, I think is great. And what our textbook does is they say, okay, here we are about to engage in metabolism, and we know redox reactions are going to be important for that. Let's dedicate an entire chapter to redox reactions themselves in general, right here at the beginning of metabolism. And that way, when we encounter the redox reactions of metabolism themselves, students will understand those reactions thoroughly because we've talked about redox reactions already, and we will follow that lead. The whole purpose of this lecture, lecture 9, is to introduce you to and get you comfortable with redox reactions in general, so that when we begin to encounter those reactions in a couple lectures as we talk about the true metabolism of glucose, it's much more of a, oh, I remember that, I know how that works. So that's really what we're going for here. But before any of that, let's get some standard definitions clear for ourselves, because this will also become important as we begin to discuss the energetics of metabolism. And this discussion pretty much revolves around what is referred to as standard states. Now, when we're doing an experiment, we, as experimenters, can define for that experiment whatever standard conditions we're using. Those can be standard conditions for some process we're using, standard conditions for the experiment itself, for the reaction. Essentially, what we mean by that is we're just defining, for anybody who's interested in our work, what temperature we were doing these experiments at, what atmospheric pressure we were doing them at, the concentration of reactants, the pH we were at, all the other parameters. So in other words, if we were going to explain a series of experiments to some reader or someone interested in our work, we would say, here is the definition of our standard conditions. Everything was done at 37 degrees Celsius. Everything was done at 1 atmosphere pressure. All the concentrations were 100 millimolar. pH was neutral at 7. And then we never have to say that again, because the reader understands that all of the reactions we describe from that point forward were done under those conditions. And they're arbitrary, right? When we define them for ourselves, we can define them as anything. We just say, this is how we did our experiments. If you want to repeat our work, you should have the same conditions. People pick it. You decide for yourself. What's important with standard states is that they are universally decided, and that chemists have gotten together, scientists have gotten together and said, okay, these are the standard states that we will all be using. So now instead of each of us describing in our papers and in our work how we did the experiments, let's agree now at the beginning that we will all do our experiments at 30 degrees Celsius, 1 atmosphere of pressure. All the concentrations we deal with will be 1 molar. So everybody uses the same standards. You can deviate from those standards if you want, but then you have to make special note of it in your description of your work. You could say that this experiment was done according to the standard states, except my solutions are at 100 millimolar, not 1 molar. So again, it's a consensus definition. And the idea of this is that if everybody's using the same conditions, if everybody's doing experiments the same way, everything is comparable. A researcher in Denver can compare their results to my research that I did here in Massachusetts, and we don't have to worry about differences in temperature or pressure because we agreed at the head of time that we were going to use the standard states. So many of you might not even remember high-definition DVDs, but for quite a while, if you wanted to watch a DVD in high definition, you could rent an HD DVD or buy a high-definition DVD, and you could watch that on an HD DVD player. But of course, at the same time, there was also Blu-ray, which is still around. And so Blu-ray was also high-definition DVD. And if you wanted to watch a Blu-ray movie, fine, but then you needed a Blu-ray player. So for a while, and by a while I mean about 4 or 5 years, half the people you knew had HD DVD players, half the people you knew had Blu-ray players. Blu-ray players couldn't play HD DVDs, HD DVD players couldn't play Blu-ray discs, and it was a big hot mess. So finally, companies decided, we got to pick one. This is insane. And they obviously picked Blu-ray. Blu-ray became the standard state of high-definition DVDs. It became the standard condition. Everyone agreed we're going ahead with Blu-ray. People stopped making HD DVD discs. People stopped making HD DVD players. We agreed on a standard. So it's the same thing here on experimental conditions. So when reactions are being done under these standard conditions, each individual condition, each individual component of that reaction is said to be in its standard state. So for pure solids and pure liquids, when we're dealing with elements, the very purity, the 100% elemental purity of those substances is its standard state. For gases, we have to deal with pressure as well. So for gases, the standard state is 1 atmosphere of pressure. For solutions, the standard state is 1 molar concentration of solutions. That's the agreed upon standard state. And you may have encountered this already, I'm sure, and perhaps you knew what it was, but maybe you didn't. Whenever you see what's called a superscript 0, like a degree sign, on any chemical term, like a delta G term or a delta E term, that superscript 0 is telling you that those numbers were measured when the experiments were done under standard conditions, that everything was done under the standard state. So again, if you saw its delta G symbolized in this way, delta G superscript 0, that's telling you that the value for that free energy change was measured during an experiment that was done under the standard conditions. So there's a problem. First of all, I'll confess a relatively large amount of disdain for the chemical field in general. I have some friends that are chemists, but really not a big fan of chemistry and not necessarily a fan of the random chemists that I should happen to meet on the street. There's a lot of reasons for that. Some of those are a little bit personal, but some of it's professional as well. Sometimes it seems to me that chemists hate biologists, and again, I'll confess to you that sometimes the feeling is mutual. Let's not get away from that. But if you think about it, the standard state that I just defined for you, now that's the standard state of chemistry, that's the standard state that chemists came up with. That standard state includes that all solutions should be at 1 molar concentration. All solids elemental, all gases at one atmosphere, etc., etc. Anybody see a problem with that when we start to think about the life sciences? In life sciences, 1 molar concentration is really, really concentrated. I mean, it's extremely rare that you would ever find anything at a 1 molar concentration in a living cell. We deal with concentrations like 1 millimolar, maybe 10 millimolar, sometimes even in the micromolar range, but 1 molar? Come on. And to give you an example, if we were dealing with a pH question under these standard states, in other words, if we had a free proton concentration at the standard state, if we had a free proton concentration of 1 molar, the pH of that solution would be 0. 0. 0 pH could, like, eat through the wall, eat through the floor. There's no way that's going to be conducive to life. So these standard states that chemistry has decided upon really don't do it for us. They are not suitable to living things. So, we're scientists, right? We can do what we want. We define our own standard state, a standard state for the life sciences. Now, to be fair, the chemistry standard state has been around for a long, long time, and a lot of very foundational research has been done using these standard states. So if we deviated from that too much, if we got a little too greedy and full of ourselves, then chemistry and biochemistry would be doing their experiments so differently we couldn't even really learn much from each other. So what we've decided as biochemists is say, okay, we'll deal with the elemental purity, no problem. 1 atmosphere of pressure works for us. The real kicker is this concentration issue, especially when it comes to free proton concentration. So what life scientists, biochemists, decided to do was allow the biochemistry standard state to deviate from the standard state of chemistry only under the parameter of pH. The standard state for biochemical reactions is a pH of 7, not a pH of 0. We've adopted everything else. Now, because we adopted a relatively modified standard state for biochemistry, we have to use a different symbol to show that as well. And the symbol we have selected is a prime, a single quotation mark. So if, for example, you were to see a symbol like this, delta G superscript 0 prime, or said more quickly and more informally, delta G 0 prime, that would tell you that the experiment that was conducted to measure this change in free energy, this delta G, was done at the biochemist's standard state, which is all of the chemistry standard state, with the exception that the pH of the reaction that was done to measure this change in free energy was a pH of 7. So with that kind of in our back pocket, that general introduction to standard state, let's change gears completely and let's talk about the big picture of metabolism. If you've ever seen the movie Jaws, there's a very good character in that movie who says that all sharks do is eat, swim, and make baby sharks. And that's very true. Sharks are incredibly efficient animals, and that's all they do. They swim around, they kill and eat things, and they make baby sharks. They've been doing it for millions of years, and they're very successful at it. If you have children yourself, or you know anybody who's had young kids not too long ago, you can say too that infants eat, poop, cry, and sleep. And man, oh man, is that true. This is Charlotte, my first daughter, and all she did was eat, poop, cry, and sleep. And she did a lot of all of it for quite a while. Both of these statements are true, and both of these statements are true pretty much of everything but everything including sharks and infants. Incidentally, 10 years makes a big difference. This is Charlotte now. 10 years doesn't seem like that much to me, but it certainly seems to have changed her quite a bit. So if you think about it, all living things, all they really do is eat, poop, essentially, procreate, and that's it. All of our behaviors, everything we do, kind of revolves around these core activities. I'll leave the last of the list to your own imagination and to other courses, but we will be considering for the next 5 or 6 weeks eating and pooping. Well, that's a little bit childish. We'll be doing it from the biochemical perspective, I guess. So eating and pooping. Well, let's make it a little more formal. Taking in materials that we need to survive, and then getting rid of the byproducts from that we don't need anymore. That sounds a little more professional, I think. Well, let's clean it up even more still. Let's make it scientific. Building the biomolecules that we require for life, and then disposing of the unwanted, unusable, metabolic byproducts that could be harmful to a cell. Essentially, that's eating and pooping, but from the biochemical perspective. It's all the same thing. We're just saying it in different ways. Don't ever forget that. Don't ever lose sight of the realness of what we're discussing because of the terminology and the technicalities that we use. So these processes, consuming food, breaking that food down into building blocks, using those building blocks and the energy to build the molecules we need, and getting rid of the waste, that is what we mean by metabolism. All of those processes taken together is metabolism. And you can realize probably already, because we started off with eating and pooping, that metabolism is made up of two large and largely separable branches. There's the eating side, that is the breaking down of large molecules that we have consumed, breaking down the proteins, the complex carbohydrates, breaking down the fats into their individual building blocks, and reaping the energy from those foods as well. All of that makes up the first branch of metabolism, the eating branch, and that's called catabolism. Then we take that energy that we have harvested and apply it to the building blocks we have eaten, and use that energy in those building blocks to build the molecules we need. See, when we eat a plant, we are consuming millions and millions of plant proteins. But here's the news. You're not a plant, and you don't need plant proteins. You need proteins. You need human proteins. So what you do is you eat those plant proteins, you digest them down to their basic building blocks, you digest them down to their individual amino acids, and then you use those amino acids to build the proteins you need. So that second branch of metabolism, the building molecules that you need, is called anabolism. So catabolism breaks things down and harvests energy. Anabolism builds molecules up and uses energy. This table from your textbook sums it up pretty nicely. We have catabolism on the left and anabolism on the right. Catabolism is taking in large molecules, breaking them down into small molecules, feeding some of them to the building processes, anabolism, and pooping out the rest. Anabolism is taking those building blocks and the energy we harvest from metabolism and using both to build the molecules we need. Under catabolism, you also see energy releasing. So harvesting energy from our foods is part of catabolism. Under anabolism, you see energy requiring. So building molecules, not surprisingly, takes energy. Also under catabolism, you see it's an oxidative process. Ooh, now that's important. Oxidation is the harvesting of electrons, and within those electrons is energy. So we get energy from food by stealing electrons from those foods. On the other side, under anabolism, we see it's a reductive process. Reduction reactions are the addition of electrons to molecules, and when you add electrons, you add energy. So we see here that redox reactions, those are reduction-oxidation reactions, are the backbone of metabolism. By stealing and adding and shuttling electrons, we are stealing and adding and shuttling energy, and that's what metabolism is all about. Also, do know that catabolism and anabolism are not the reverse of each other. If catabolism is the breakdown of biomolecules, it does not include the breakdown of our own biomolecules. That would kind of be the infinite loop of we make a protein, we digest it. We make a protein, we digest it. No. Here we are taking external proteins from the foods we eat, breaking them down, and then using them to build something new through anabolism. So just to review, catabolism is when we break things down and release energy. That's at the heart of eating. That's why we get food, why we eat food. We get food and eat food in order to get those building blocks and that energy. It's an oxidative process. It requires the stealing of electrons. Anabolism is the building side of metabolism when we build molecules that we need to sustain our own life Of course, if we're building molecules, we're decreasing entropy, we're increasing order. That means energy is needed. Anytime we build anything, energy is going to be needed because it decreases randomness. This is a reductive process. It requires electrons. When we feed electrons into these processes, we're feeding energy in as well. And so these redox reactions are essentially oxidation-reduction reactions that involve the transfer of electrons as a means of transferring energy. And that is the crux of redox reactions as far as metabolism goes, and it's why we're spending so much time on it here. The way that we move energy around from one molecule to another, the way that we get energy from catabolism and feed energy to anabolism is by shuttling electrons around the cell. So, to summarize what we talked about in this chunk, we started off by talking about the standard states and standard conditions, symbolized by a zero, a superscript 0, a degree sign. So if you see something like delta G superscript 0, it's telling you that the value for that free energy change was calculated by measuring a reaction that was done under standard conditions, and we define some of those. Because the standard conditions includes a molarity of 1 molar, and that really doesn't cut it as far as pH goes, biochemists have defined their new standard state, delta G0 prime, where we've adopted all of the standard conditions of the typical chemist's reactions, but we've changed the pH from 0 to 7, something much more conducive to life. From there we moved on to metabolism. Breaking down what we eat, building up what we need, and disposing of the waste is metabolism, eating and pooping. We broke that down to catabolism, the breaking down part, and anabolism, the making part. Oops, sorry about that animation there. As we build new molecules, we are engaged in anabolism, and the way that we feed energy into anabolism and extract energy from catabolic reactions is through redox reactions, by shuttling electrons around. So that's where we'll pick up in the next chunk. We'll talk quite a bit about redox reactions in general so that you're comfortable with them as we move on to metabolism.